So this lecture is to help walk you through pedigree analysis. This is chapter 6 in your textbook, and we're going to learn about pedigrees, and we use these, you've heard about them for dogs. Oops, I forgot to turn on my pen again. And horses and other kinds of animals. And we use them to look at inheritance of humans because humans are a little more challenging. One, we don't usually have lots of offspring to help us determine how a trait is being inherited. Unfortunately, we can't control breeding in humans, right? So we can't do things like, let's breed this guy and this girl and see what their children look like and see if we can figure out, how freckles are inherited. So the classical way is to look at these things called pedigrees, shown down here. So a couple things about pedigrees. Understand that males are always designated as a square and females as a circle. And a line between them means mating. And you can follow, if you come over here to the pedigree, you go down and here's the next generation, and everyone that is connected to these two parents are the children. Right? And then people that are not connected, these are the people that are coming into the family and breeding and having more children. When the square or circle is faded in, that means that person is affected. Okay, and affected can mean anything from, if you're looking at the inheritance of freckles, has freckles, or has a specific disease, say. So affected means you have it. It doesn't tell you yet how that gene is inherited. Some pedigrees will put a slash and fill in half the circle or half the square. These people are designated as a carrier, which means they're heterozygous. But many pedigrees do not make that designation. And like you can see in this one, right, you can be heterozygous. This is a dominant trait. So a carrier would be designated for a recessive trait. And carrier, though it means heterozygous, it also means unaffected. Right? And this is where people are most curious, right? You might carry the gene for a certain trait and not know it because you do not display the phenotype. We designate the generations by Roman numerals, and we designate the people in the pedigree along one generation by numbering 1 through whatever. Okay, so this is a way you can say, okay, person 3, generation 2, so 2-3, we can talk about their genotype or their phenotype. Again, you have to look at a pedigree as a whole to determine dominant, recessive. And those are the only kinds of pedigrees we're going to work with. We're going to work with dominant and recessive autosomal inheritance, dominant recessive X-linked, and might even throw in a Y-linked inheritance. All right, so let's look at the 5 types of pedigrees you need to be able to recognize and how you do that. So this first one is autosomal recessive. Okay, so that means affected people will be homozygous recessive. So, looking at this pedigree, if you knew it was autosomal recessive, you could start filling in the genotypes for some of these people. But the big question is, how would I know this is autosomal recessive? What you want to look for is an affected child with two unaffected parents. Okay. So I will tell you right now, any autosomal recessive pedigree I give you will have this characteristic. And so if you look through this pedigree, I usually start at the bottom and go up. You can see that this child has two unaffected parents. And the reason that's so key is because it's telling you that these parents were both carriers, and it's kind of like, whoa, all of a sudden this trait popped up. We weren't expecting it, right? Because they were both unaffected. I'm trying to... Some other things, and there's a table 6.1 in your text that lists a bunch of, a bunch of characteristics for looking at pedigrees. There's no way you can remember all of these, so I'm going to give you the ones that stand out the most. Excuse me. So you'll also see things, because you might say, well, is it X-linked recessive? So you'll see males and females in equal portions, and fathers that can pass on the trait. Do we have one? Fathers that can pass on the trait to their sons. Some of the ways to determine if it's autosomal or X-linked. If you get a problem that says it's a rare allele, then you assume that people outside of the family, so those coming in to breed, you like that? Breed. Do not have or carry the trait, the allele, sorry. Assume that people outside the family do not have the allele. Oops, too low on my screen. Do not have the allele. Okay. Many pedigrees will fit autosomal recessive. It's very easy to make them work. So I will not accept autosomal recessive unless you have something like this, where you have an affected child with two unaffected parents. So just because it can work doesn't mean that is truly the mode of inheritance. Okay, let's look at autosomal dominant. Oh, let me go back here. And here you can see that, let me get rid of some of this coloring. These people you could fill in as carriers, right? So they carry the the recessive allele, but they don't show it. But many of the pedigrees I will give you do not show that people are carriers. Okay. Autosomal dominant. So what do you know about this? You know that it is caused by at least one dominant allele. Right? So if you started filling in this pedigree, you would know that everybody that is affected must have a dominant allele. On the flip side, everyone that's non-affected must be homozygous recessive, because dominant dominates, right? And this one can become pretty easy to figure out everybody's, or many of the parents' alleles. For instance, we know that these parents must be heterozygous because they had a homozygous recessive child. But for these three right here, you can't assign full genotypes because you don't know. Well, this one you can. Sorry. I'm wrong. Ah, darn it. These two, you don't know if they're carrying two dominant alleles or one dominant and one recessive allele. Okay. So what are you looking for autosomal dominant? Okay. Again, you see similar ratios in the sexes. The key is every affected child must have at least one affected parent. Right? So it can't be a surprise because it's dominant, so the parents are going to show. So again, in this example of autosomal recessive, this would tell you it's not dominant because you have an affected child with two unaffected parents. So again, I always start at the bottom and start looking up to compare child with what their parents have. X-linked recessive. Okay. A couple things to note about X-linked recessive is it's more frequent in males, right? Because an X-linked recessive male, because all males only have one X chromosome, they will show the trait more often than females. So females have to be homozygous recessive. Important is it is not passed from father to son. This is a key to help you determining X-linked versus autosomal. Dads contribute the Y chromosome. We are talking about X-linked chromosomes. Okay, so it cannot be passed from an affected father to a son. Heterozygous women, okay, in theory, could have half normal, half affected sons. But remember that people don't usually have enough kids to show nice Mendelian inheritance. So you have to do a little analysis. The other thing that, since it's not passed from father to son, is that any affected female must have an affected father, because she has to have two recessive alleles. For X-linked dominant traits, where the affected people are of genotype X capital N something, or X capital N Y, again, you see that every affected child must have an affected parent. This isn't one that hides out. Again, it cannot be passed father to son, so that's very different than autosomal dominant. Like I said, every affected child has to have at least one affected parent. And affected men, as shown right here, will pass it on to all daughters. Okay. So daughters get an X from their mom and an X from their dad. If the dad could be an X, if the dad has a dominant allele, then all his daughters will also be affected. And then finally we have Y-linked, where finally you can pass it on from dad to son, right? And that's the only way, because moms don't have a Y chromosome, girls don't have a Y chromosome. So Y-linked is pretty obvious. So let's look at a pedigree and figure out how to tackle it. Okay. So given this pedigree, how is this trait inherited? And I'm going to walk you through what I do. And again, I always start at the bottom and work my way up. And I see, okay, I have a couple affected kids, and wow, no affected parents. So I know it has to be a recessive trait. And because the dad is not affected, but the daughter is, it has to be autosomal. So let me show you that. So in this case, the dad and mom are both carriers, right? And they produce these affected children. If you were thinking it might be X-linked, so that the daughter was this, she would have had to have an affected father. So I can tell immediately it's not X-linked, and it's autosomal recessive. So then you might be asked to fill in genotypes, and you could work through, and again, lots of people with autosomal recessive traits, we can't tell their exact genotypes, so the correct way to represent them is a capital letter and a dash. So why don't we know all the genotypes? Because big A, and big A, little a are going to give the same unaffected phenotype. One of the other things we can do with pedigrees, in addition to deciding how something is inherited, is we can do probability. And this is where things like genetic counseling would come in. That they would look at your family pedigree for this disease, say, and they would predict your chance. So it's a little more complicated than Punnett squares, because we're assuming we don't know exactly what the genotype of the parents are. Now you can start to figure out genotypes of the parents if you do some molecular biology and DNA sequencing. But we're not there yet. We're still kind of on those classical genetics. So my question to you is, if individuals 3-1 and 3-7 decided to have children, what is the probability that they will have an affected child? So what you first have to figure out, okay, is that the only way for them to have an affected child is for them to be both carriers. So you first have to decide the probability that they are heterozygous. They are heterozygous. So we look at, 3A's parents. Big A, big A, big A, little a. So what's the probability of her being big A, little a? We've done these kind of probabilities before, right? Probability is half. 50-50. And we look over at person 3-7, and we look at their parental genotypes, and it's the same probability, 1/2. Right? So we've got that it's 1/2 chance that the mom is heterozygous, 1/2 chance that the dad is, and then we know from a monohybrid cross the chance of producing an affected child is 1/4. So we multiply all these probabilities together and get 1/16. Okay, so you'll be asked to do some problems like this. Let's look at a different one. Same pedigree. And now, unfortunately, we're going to pretend these people were separated early on, didn't know each other, decided to have children together. Right? Just for hypothetical sake. Okay. So, again, these parents both have to be heterozygous to have an affected child. The probabilities are different here because if you look at their parents were both heterozygous. So we know that you get a 1:2:1 genotypic ratio. Since these people are not affected, the probability that they are heterozygous is 2/3. This is another conditional probability. It's not 1/2 because we know they're not affected. So we remove that from the probability. So the probability that each of these people are carriers is 2/3. The probability of producing an affected child is 1/4. And so if you multiply these all, you get the probability of having an affected child as 1/9. And honestly, this is why in some populations, some small populations, recessive traits seem to show up. Let me just say, instead of just recessive, but recessive traits that could have a negative impact show up more frequently because of small breeding groups, right? So brother and sister can be carriers and have a higher chance of being a carrier, and they're going to therefore have a higher chance of producing a child affected with whatever we're looking at. So small populations keep these genes at higher these alleles at higher levels because there's less genetic diversity in their group. So that's how you tackle pedigree problems. Okay? Those are the types of questions you can be asked to identify the type of inheritance, the genotypes of different individuals, and you can be asked to do probability problems. So there are 4 clicker questions here. I have posted in this module, week 4, these pedigrees individually as slides, so if you want to print them out bigger and work through them. There is also an extra credit option where you are going to draw the pedigree. So it's described here on this slide, and then this is what you need to do. You get 15 points added to your class participation, so if you've been missing some clicker questions, or you've been missing class, or you didn't have your clicker, this is a great way to boost that grade. Due at the beginning of class on Thursday, no exceptions. Write it neatly, does not have to be typed. I want you to draw the pedigree as described, number the generations and the individuals, assign genotypes, and then what is the probability that both twin daughters are carriers? And that comes out in this part. All right, have fun with pedigrees. Thanks.